How's everybody doing? How was the exam? Sorry to bring up a bad subject. 5 more minutes. Was time a factor? Was it? Okay. There were longer answers. That probably was part of it, how long it took you to write something, I imagine, yeah. Other thoughts? It's not graded yet, so I can tell you that. Just as hard as the last one, not more difficult? Hard as the first. Okay. So it's probably harder than the last one is what you're saying. Okay. Okay. Was it representative of the material or not? Maybe? Okay. Okay. All right, well, we are in the home stretch now. So, as I said last time, for the final exam, you will get a note card. You have to get the note card from me, and you'll be required to bring a note card. That's another thing. So you have to get it from me, and you have to bring it. And if you don't bring it, you'll lose points. And there's a reason for doing that. I don't do it to be mean, but I want everybody to fill out their own note card. And you say, well, duh, I'm going to fill out my own note card. And you will. But what happens is if you don't turn it in to me, then you pass it off to your friend next year. And your friend isn't going to benefit from making the card. So that way I keep track of things. If you want to photocopy your card, that's fine. Go ahead and do that. But they still have to get the card from me, just like you guys have to get the card from me. So that's the reason that I do that. And what you will find in the process of making that card is, if you haven't done this before, you'll find it's a very good tool for organizing information. And you will find that you won't use it nearly as much as you think you will. And you're welcome to use it, of course. But I think you'll find that you'll be much more organized in your head as a result of organizing the stuff on that card. So I hope that's the case for you. It's a big 5x8 card. Yeah. I can make it smaller. I mean, if that's too big, I mean, we could reduce it in size and everything. So he wants to bring a magnifying glass. The best scheme I ever saw was I had a student one year who said, can I write in 2 different colors of ink? And I said, sure. And then he said, can I bring 2 different colors of glasses, like the 3D glasses, one red, one green? And so at the time I said, sure. And then I discovered what he did. It was amazing. He had one, when he wanted to read the red stuff, he put the green one on. And when he wanted to read the green one, he'd read the red stuff. So he actually had twice as much information on the card as you could have gotten in otherwise. So I subsequently banned coloring lenses. I don't have a problem if you want to. Now, the other thing is you have to completely write it in your own hands. So you can't print something, you can't photocopy something. You've got to print it with in your own hands. But if you want to print something in your own hands and bring a magnifying glass in, I don't have a problem with that. If you can print that tiny, yeah. I do respect intelligence, absolutely, yeah. Well, I respect intelligence, but I also have to be careful with giving someone an advantage over others. And so fairness is an important consideration. So that was why I did that. But I thought it was intelligent. I thought it was clever. It was a very clever idea. So, okay. So as I said, we're moving along. We are at that pace where right now we're going to be going through about a chapter a day. So it's going to be fairly quick-paced the rest of the way on out. And again, I will try to emphasize the highlights for you as we go through, hit the bigger landmarks as we're going through and doing that. So the first thing we're going to talk about is central metabolic, is a citric acid cycle, which is a central metabolic pathway. So I described glucose, boy, my mind is not working, glycolysis as a central metabolic pathway. Similarly, the citric acid cycle is a central metabolic pathway, meaning that we find it in essentially all organisms. There are a few organisms that have some variations on this, but essentially we find this in all organisms. And the reason we do is that it is so central to the cell. What you'll see as we get going through this is that there's many things that are in this cycle that can be used to make other things, So we can use some of the intermediates to make amino acids. And conversely, we can use amino acids to make intermediates in this cycle as well. So it's central because it's very integrated with other pathways that occur inside of the cell. So if we look at the overall scheme looks something like that. And what you've seen so far has been this, glucose going to pyruvate. Today we're going to talk about pyruvate going to acetyl-CoA and then acetyl-CoA entering the citric acid cycle. But acetyl-CoA itself turns out to be a fairly central molecule in a lot of different metabolic pathways. Fatty acids break down to acetyl-CoA. Amino acids break down to acetyl-CoA. So, and acetyl-CoA, as I said, is the entering material for the citric acid cycle. So the cycles or the pathways really do interconnect. And that's why I say we can't really say this is part of one pathway, this is part of a different pathway. Pathways really are man-made inventions, but they do help us to organize how we think about things. This cycle is the most oxidative cycle that, or I shouldn't say the most, but it probably is the most oxidative cycle we find inside of cells. Fatty acid oxidation would be the only thing that would be even close to it in terms of the amount of oxidation that's occurring. Okay? So a lot of oxidation occurring here, meaning that we produce a lot of NADH and we produce a lot of FADH2. Okay? So most of the energy of glycolysis is realized in this pathway because of the numerous oxidations that actually occur. Okay. The citric acid cycle occurs exclusively in the mitochondrion. And that means it's appropriate we talk a little bit about mitochondrial structure. Okay? So mitochondrial structure, mitochondria, of course, are the organelles that we refer to as the power plants of our cells. They're an organelle of eukaryotic cells, and they're the place where the vast majority of cellular ATP is made. The mitochondrion has a few structures that you should be aware of. So first of all, it has a membrane going around it we call the outer mitochondrial membrane. And the outer mitochondrial membrane doesn't have a very important function in the overall process. The inner mitochondrial membrane, however, has enormous importance and enormous functions in the processes we're going to be talking about over the next few lectures. Okay? So the inner mitochondrial membrane actually has embedded in it one of the enzymes of the citric acid cycle. And the rest of the enzymes of the citric acid cycle are dissolved in this central component called the matrix. So the matrix is simply the equivalent of the cytoplasm of the mitochondrion. It's just the liquid, free component of the mitochondrion. All right? So it's in the matrix where the other 7 enzymes of the citric acid cycle are found. So all of them are found in the mitochondrion, one's in the inner membrane, 7 of them are dissolved in the matrix. Now we'll talk about electron transport and stuff later, so I'm not going to go through that here today. Oh, and there's also this intermembrane space. The intermembrane space does play a little bit of a role later when we talk about electron transport, and I'll talk about that at that time. Okay. This figure is a nice overview of the pathway of electron transport. So I want to say a few words about that. And then I'll go through some of the considerations of the individual reactions. So this pathway is a different one than you saw of glycolysis. Glycolysis we could look at and say it had a beginning and it had an end. This guy doesn't have a beginning and it doesn't have an end. Okay? It is a cycle, which is why we call it the citric acid cycle. You also hear this called the tricarboxylic acid cycle or TCA cycle, some people call it. And some people call it the Krebs cycle. So all those names are equivalent. They all mean the same thing. But you can probably call it the citric acid cycle since that's what most people call it today. Okay. Now, this cycle is important because, as I said, it's one of the major oxidative pathways of the cell. The cycle starts with entry of acetyl-CoA into it. And the acetyl-CoA is joined to this molecule called oxaloacetate. And you heard that molecule last time when I talked about gluconeogenesis. And you may remember that I said in gluconeogenesis, that reaction that made oxaloacetate was the only one that occurred in the mitochondrion. So, and here's oxaloacetate in the mitochondrion. So that's kind of an interesting connection there. This 4-carbon oxaloacetate can be combined with the 2 carbons of acetyl-CoA to make a 6-carbon molecule. And then that 6-carbon molecule, I'm just stepping you through it quickly now, I'll do the individual reactions in a second. The 6-carbon molecule called citrate or citric acid is rearranged. So it's just an isomerization that occurs. Then there's an oxidation that occurs with loss of a carbon dioxide. That's a decarboxylation is what it's called. There's a second decarboxylation that converts that 5-carbon intermediate into a 4-carbon intermediate linked to CoA. Then the 4-carbon molecule is released from the CoA. Then there's an oxidation. It's an unusual one we'll talk about. And then there's addition of water. Finally, yet another oxidation, and then we're back where we started. Now, the fact that this pathway ends up with what it starts with has importance. Right? Well, obviously, we wouldn't have a cycle if it didn't end up with what we started with. But this oxaloacetate that we see here is an important consideration. You'll notice that one turn of the cycle, we don't have any more oxaloacetates than what we started with. We have exactly the same number we started with, which is 1. We start with 1, we end with 1. We added 2 carbons, we lost 2 carbons to decarboxylation. So what that means is we have no net production of oxaloacetate. No net production of oxaloacetate. Now, that won't have much meaning for you at this time, but I want you to plant that in your memory because as dietitians, for example, that has great importance because it means that you can't use the citric acid cycle to make glucose. You can't use the citric acid cycle to make glucose. We'll see why that's important later. But I'm just planting that idea in your head right now. Okay. Now, to get to the citric acid cycle, we've been following the path from glucose. So I want to continue that path from glucose into the citric acid cycle. As I noted earlier, there are 2 different ways that we can bring, or 3 different ways we can bring acetyl-CoA in, by glucose, by fatty acids, and by amino acids. All right, we're going to follow the pathway in from glucose, but it doesn't have to be that pathway. So I'm just going to show you how it goes from glucose. Well, pyruvate in a reaction that occurs in the mitochondrion. This reaction you see on the screen occurs in the mitochondrion. Pyruvate can be decarboxylated, meaning that it can have one of its carbons chopped off. It can have a CoA attached to it. That's what makes acetyl-CoA. And, more importantly, it produces NADH. All right? This is an oxidation that's occurring in this process. We talked about it before, and I'm just sort of showing you something you saw in the last exam. This reaction, as I said, occurs in all organisms that are aerobic, and it occurs quite readily. And it occurs when oxygen is present. When oxygen is present, that means we have plenty of NAD. When oxygen is present, underline that in your heads, it means we have plenty of NAD. Okay? So when oxygen is abundant, we're going through decarboxylating pyruvate, making acetyl-CoA. Okay? All right. Now, this reaction is catalyzed by something called the pyruvate dehydrogenase complex. And this was the reaction that required these several vitamin-related cofactors. One was TPP. Second was lipoic acid. The third was FAD. The fourth was CoA. And a fifth was NAD. They're all on the screen there. 1, 2, 3, 4, 5. All 5 of those are necessary for this reaction. That says that this reaction, A, is kind of complicated, and it is. We're not going to go through the mechanism of it, so you don't have to worry about that. But the fact that there are 5 things there tells us there's several things that have to happen. Okay? And those things that have to happen include not only the decarboxylation, but also an oxidation that occurs. And this is a multi-subunit enzyme, and these different reactions move through different parts of the enzyme. So that's why some of the other molecules are necessary. They're necessary for carrying the molecule as it's going through these reactions. Okay. Now, as I said, I'm not going to hold you responsible for mechanism. There is the mechanism if you'd like to see something about how it goes. I'm only showing you this figure to show you that some of these things, like lipoic acid right here, are carrying, okay, the intermediate during the oxidation process. Here's TPP carrying the intermediate during the process. So during the oxidative process, it's occurring inside this enzyme. As I said, there's different places in the enzyme where the reactions are occurring. So to make sure the molecule doesn't go flying away, these carriers actually carry it from one place to the next. The overall upshot of this is that NADH is produced, acetyl-CoA is produced, and carbon dioxide is produced. All right. Well, acetyl-CoA, excuse me, acetyl-CoA is the thing that we are most interested in understanding. And acetyl-CoA is important because it's the first reaction, or I shouldn't say first, because we have a cycle. There's really not a beginning and an end point. But almost always we refer to this reaction of oxaloacetate plus acetyl-CoA as the first reaction. Okay? We could literally define the first reaction anywhere in there, but we're going to call this one the first reaction. Well, if we look at that reaction, here's what's actually going on. Oxaloacetate, 4-carbon molecule. No, you're not going to need to know the structures here. I do think you should know the names of the intermediates. I think you should know how many carbons are in the intermediates. But I'm not even going to hold you responsible for enzymes. How's that? Okay? Names of the intermediates, number of carbons of the intermediates, but I'm not going to hold you responsible for the enzyme names. All right. Now, 4 carbons plus 2 carbons. Those 2 carbons, oh, that should be a C. That's an O, and I just discovered that. So it should be a CH3 right there. All right. CH3 gives us a 6-carbon intermediate that looks like this. Now, the 6-carbon intermediate is citrate. That's why the enzyme name is citrate synthase. Again, I don't care if you know the name or not, but the important thing is we've made a 6-carbon intermediate. We've brought in 2 brand new carbons for the enzyme to use. Notice in the process that coenzyme A, CoA, is released. All right? Now, there's something else about this reaction that's important, and that is that you notice it doesn't take any ATP to put these pieces together. That's a little unusual. A little unusual. Okay? It actually takes energy to put pieces together like this, but you don't see any ATP energy. And the reason you don't see ATP is because acetyl-CoA is an activated intermediate. Does anybody remember what that is? Anybody remember what an activated intermediate is? Energy stored in its bond. That's a simple way of putting it, but right. So this guy has energy stored in this bond between this sulfur and this carbon. And the result of that is that when you break that bond to attach this guy up here, then that means that my mind's not working at the moment. That means that the energy released by breaking that bond can be used to make this structure up here. And it turns out that the energy in that bond is pretty significant. It's a pretty fair amount of energy that's there. Okay? Okay, and we'll see evidence of that later. Yes, sir? Yes? That O right there should be a C. Yes. I'll change that tonight. I didn't realize that was there. That should be a C. Sometimes, as I told somebody, you look at these for a long time and you think you've seen them, because I had students draw these figures, and I thought I'd gotten all the errors, but that looks, when you're just glancing at it, you don't notice it until you're standing in front of a group of people like I am right now, and there it is. That should be a C, but I will change that. Okay, so there's the first reaction of the pathway. We've made a 6-carbon intermediate. The second reaction of the pathway, as I said, is an isomerization, meaning that we're just rearranging the components of the molecule. Citrate is becoming isocitrate. All right? Now, it's a pretty minor reaction. It's not a biggie. This is, well, one difference is this is a symmetric molecule. This is an asymmetric molecule because this guy has, though it has, this only has 3 different things attached to it because this thing is equivalent to this thing. All right? But down here, now this carbon has 4 different things attached to it. This is not equivalent to this, that, and that are all different. So we've made an asymmetric molecule in the process. The enzyme that catalyzes it called a aconitase, and this is one enzyme I'm going to ask you to know something about. Okay? One enzyme. And I'll tell you why. It's actually on the next slide, if I can show you that. It's an interesting story. There's a compound called Fluoro Citrate that was used back in the 1960s as one of better ways of living through chemistry. And the idea of better living through chemistry back in the 60s was that, one of the ideas was that we could use chemicals to make our world a better place. That's how DDT came into being and so forth. Well, this guy that you see on the screen had a pretty big effect, kind of like DDT did as well. And the reason was as follows. Fluoro Citrate, first of all, can go and be combined. The aconitase enzyme looks at this, the citrate synthase enzyme looks at this and says, that's just the same thing as acetate. So we make some acetyl-CoA. This is a, let me start over again. All right, I've got fluoroacetate. The cell looks at this and says, I can put a CoA on there and make fluoroacetyl-CoA. Citrate synthase says, oh, that's the same as acetyl-CoA, so I'll make fluorocitrate. Okay? And it does that. And that's fine. That has no problem, no effects whatsoever. Fluorocitrate, however, kills aconitase, the next enzyme. So this is an example of what was described as a suicide inhibitor because this kills the next enzyme in the cycle. This cell is actually putting something together that's going to kill itself. You can't live without a functional citric acid cycle. Well, we didn't give this to people, okay, because we weren't in the business of killing people other than by war, the traditional means, okay. Instead, we said, well, this would be a great way to kill coyotes because coyotes and wolves and other animals that were out on the prairie that were killing livestock were causing a lot of economic harm. And as a consequence, there was a lot of interest in reducing their populations and or killing them. So they started putting out fluorocitrate out for coyotes to eat and so forth and to kill them. And it worked very well, and it killed them. And the thinking was that, well, this isn't going to be something, humans aren't going to eat coyotes, and we're not going to get this into our food chain, et cetera, et cetera. And we largely didn't get it into our food chain. Okay, that was the good news. The bad news was that the birds that ate the carcasses of the coyotes and so forth found themselves poisoned, and the bird populations, as a result of this, were decimated. Okay? Because it went through the bird food chain very well. Well, not all the birds ate coyotes, of course, but those that did passed on or didn't pass on or had to eat other things, and the bird populations in the West, as a consequence, plummeted as a result of the use of this. Well, this was eventually banned for that reason, and it happened because aconitase gets inhibited by this enzyme. So that's why I want you to know something about aconitase. All right. The next reaction is the first oxidation that occurs in the citric acid cycle, and it occurs here. And what we can see happening is here's the hydroxyl group on isocitrate that's now been placed into that bottom position. It is becoming oxidized. So when it becomes oxidized, what happens is we get a decarboxylation, meaning that we lose this carboxyl group, and we end up making something called alpha-ketoglutarate. Right? I'm pointing to the wrong carboxyl that's leaving. It's actually the middle carboxyl. It's not the end one. It's the middle carboxyl. Okay? Not that it matters. You're not going to have to know which one. But the middle carboxyl is the one that leaves. All right. Well, the upshot of that is that this alcohol becomes a ketone. That's an oxidation. And the NAD is converted to NADH. So it's a decarboxylation. It is an oxidation, both of which are occurring. We've gone from 6 carbons to 5 carbons. Now, alpha-ketoglutarate turns out to be a really important molecule in amino acid metabolism. A very important molecule in amino acid metabolism because if I replace that oxygen right there with an amine group, I have glutamic acid. So This is very easy to convert a citric acid cycle intermediate into glutamic acid. It's also very easy to convert glutamic acid into alpha ketoglutarate. Okay. Yes? Are all these reactions reversible? That's a very good question. The oxidative decarboxylations tend not to be very reversible because the CO2 sort of disappears. So the isocitrate, the aconitase reaction is essentially reversible. The glucose, citrate synthase reaction is not reversible. So they're all theoretically reversible, but in practice, the only one I've talked about so far that's very reversible is the citrate to isocitrate. Whenever you have a decarboxylation, again, it's theoretically reversible, but usually the CO2 goes away, and then it's hard to make that reaction go backwards. Okay, the next reaction, the 5-carbon intermediate of alpha ketoglutarate is converted into a 4-carbon intermediate, and we've got a couple things going on here. All right? We have NAD is used, NADH is produced. We have carbon dioxide produced, and we have CoA attached. Now, all of those things happened on an alpha keto acid. What is an alpha keto acid? Well, an alpha keto acid is an acid that has a carboxyl, and the next carbon has a keto group on it. Now, you may not have noticed when I showed it before, but pyruvate was also an alpha keto acid. All right? That means that this reaction that's occurring here is very, very similar to the reaction that occurs in the pyruvate oxidation. And it's not surprising that this reaction uses exactly the same intermediates. It uses NAD, it uses TPP, it uses lipoic acid, it uses FAD, and it uses coenzyme A. It's a different enzyme that it uses, alpha ketoglutarate dehydrogenase, but its structure is very similar to pyruvate dehydrogenase. Very, very similar. Mechanism of reaction, very, very similar. The net reaction is that we are putting a CoA onto that 4-carbon intermediate. And what I've told you before was that this guy is going to have energy, right? It had energy when we made the acetyl-CoA before. Here you go, sir. You bet. Okay. And we'll see in the next reaction that this energy is actually used to make something. Okay. Let's take a look at that. Here is succinyl-CoA. Now, this reaction is the only place where a triphosphate is made in the citric acid cycle. On the last time I talked about this in glycolysis, I didn't give you a name for that, but I'm going to give you a name for it here because it'll become important next week. All right? Whenever we make a triphosphate directly in a reaction, metabolically, whenever we make a triphosphate in a reaction directly, metabolically, all right, that process is called substrate level phosphorylation. I think my song actually talked about it. Substrate level phosphorylation. So if I take a GDP and make a GTP, I've done substrate level phosphorylation. If I take an ADP and I make an ATP, I've done substrate level phosphorylation. So anytime I'm directly making a triphosphate in a reaction like this, I'm doing substrate level phosphorylation. I tell you that because there are 3 types of phosphorylation that occur in cells. 3. This one is a relatively minor one. All right? We don't make very much of our triphosphates by substrate level phosphorylation. Very little of that. Most of our triphosphates come from what's called oxidative phosphorylation. That's what we'll talk about next week. That's what happens inside the mitochondrion. Okay? Now, these are reactions that are occurring inside the mitochondrion, but they're not the oxidative phosphorylation. We'll see how that works next week. The third type of phosphorylation that occurs inside of cells occurs only in plants and photosynthetic bacteria, and it's called photophosphorylation. And that's where the energy of light is used to make ATP. And we'll see how that occurs when we talk about photosynthesis. Okay. Let's see. So, actually, one reminder, just a reminder here, all right? Now, this had enough energy to make GTP. From GDP, that means that this guy had a lot of energy in it. You don't make a triphosphate unless you have a lot of energy input, and the energy input is coming from that CoA being linked to the succinate. All right. The next reaction is an oxidation, and I said it's a little bit of an odd oxidation. For the first time, you're seeing the electrons being donated to FAD to make FADH2, and that's because this type of an oxidation is a different type of an oxidation than you've seen before. You've seen oxidative decarboxylation in a couple of reactions. You saw another oxidation reaction with NAD and NADH back in glycolysis, but you haven't seen an oxidative reaction where electrons and protons between two carbons are being removed. This type of oxidation is a different type of oxidation than you've seen, and electrons in reactions like this are donated to FAD. There's actually an energetic reason why that happens. You don't need to know it. We won't go through it here. But there's an energetic reason why they're donated to FAD and not to NAD. The upshot of this is you make FADH2, and you get a molecule that has lost 2 protons, 2 electrons, and it has this structure that you see here. Okay? That's the structure that it has. Fumarate is a molecule that has a trans bond. And when we talk about the oxidation of fatty acids next week, okay, I think it's next week, I will remind you of this reaction because the reaction that I'm showing you is almost identical to one that occurs in fatty acid oxidation. In fact, this reaction, the next reaction, and the reaction after it all occur in fatty acid oxidation. Okay? So that means if you learn this here, you're already learning 3 of the 4 reactions that occur in fatty acid oxidation. Okay. All right, so that is our third oxidation that's happened. We've made so far 2 NADHs and 1 FADH2. The next reaction is a pretty simple one. For anybody who's had organic chemistry, you know you can add water across the double bond like this, and that occurs fairly readily. When you add that water across the double bond, you make something called L-malate. Notice it's stereoisomer because this carbon has 4 different things attached to it again. And then the next reaction is a very odd oxidation reaction. Okay? A very odd oxidation reaction. It's a very odd oxidation reaction because its delta G0 prime value is positive. Not too many oxidations that have a delta G0 prime that is positive. Well, a positive delta G0 prime reaction means that we have an energy hump we have to get over. We had an energy hump with aldolase. How did we get over the energy hump in glycolysis? 2 principles. What were they? Pushing and pulling. Okay? Well, it turns out that this guy gets pulled. It gets pulled. So what does pulling mean? That was an exam question. What does pulling mean? Take away products, okay? And taking away products occurs in the next reaction. Well, the next reaction was that very first reaction that I showed you. That was where oxaloacetate was combining with acetyl-CoA to make citrate. That reaction is very energetically favorable. It's very, very energetically favorable, meaning that there's very little oxaloacetate that's left, and this reaction gets pulled. So that's how this reaction actually goes forward in the cell, is by pulling away the products in the very first reaction of the citric acid cycle. Well, we're back where we started. We've now made our last NADH, and what we've done so far is we've made 3 NADHs, we've made 1 FADH2, and we've made 1 GTP. If we were to go all the way back to thinking about glucose, glucose gave us 2 pyruvates, right? So that means we're going to have to run this cycle twice to get all of the intermediates from glycolysis oxidized. And in fact, when we do that, what we discover is that we double all of these. So instead of 3 NADHs per each glucose, we're getting 6 more NADHs. Per each glucose, we're getting 2 more FADH2s. And per each glucose, we're getting 2 GTPs. All right? Now, we'll later determine how many overall ATPs we make, because NADH and FADH2 can both be converted into ATP. And as I've said in class before, it varies on how you count those. I count them a little bit differently than most people do. But if you count them my way, you end up with 38 ATPs per glucose if you completely oxidize glucose. 38 ATPs per glucose. Okay? That's a lot of energy. And we compare that with fermentation, which only had 2 ATPs per glucose. We see that this cycle is giving us a lot of metabolic energy. All right. Now, I'm doing well on time. And yes, I'm going to let you out a little bit early today. I want to say a couple things about the cycle. Okay? Now, here he goes about regulation again, right? Think about regulation. I'm tired of regulation. Well, we're going to see more regulation. And regulation is really where the interesting things are in these pathways. This thing looks a lot bigger and a lot hairier than it really is. And we're going to look at this one, the regulation of this pathway, in the simplest possible way. You guys like simplicity? I like simplicity. And while these various things do affect the different enzymes, ATP, NADH, acetyl-CoA, ADP, NAD, ATP, blah, 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 right? The bottom line of this pathway down here, okay, which includes the entry of the citric acid cycle, everything below acetyl-CoA, the bottom line of this pathway is that the regulation is extraordinarily simple. The primary thing that determines if the pathway goes or not is the amount of NAD that's available. Hey, that sounds just like glycolysis, right? We did fermentation so we could remake NAD to keep glycolysis going. Similarly, if we run out of NAD, or FAD for that matter, NAD or FAD, this pathway will not run. Doesn't matter about allosteric inhibitors, doesn't matter about anything else. It simply means the pathway won't run. And for 99% of the things that are happening in your cell, which is good enough for us, that determination of the NAD and FAD level is really all we need to worry about in understanding this cycle. That simplification will allow you to understand very, very well next week how your body works. You thought you understood how your body works. You're going to learn a lot more about it next week. Okay? Because this limitation of NAD and FAD availability determines a tremendous number of things. Okay? All right. Now, even this reaction up here, which I didn't refer to in the group, even this reaction requires NAD. Whenever you see it's inhibited by NADH, that means it needs NAD. Okay? So, even the entry of pyruvate into this pathway requires NAD. So NAD is the primary thing that we're going to be interested in understanding. Well, there's something you don't have to know, but I show it to you to show you the various things I was talking about. I told you that that last step was oxidatively very unfavorable. Look at that. Look at that. Delta G 0' of 29.2 positive. All right? Here's that reaction that was pulling it right here. Minus 32.2. They basically balance out. The overall delta G 0' for all the reactions is minus 77, meaning that this overall process is very, very energetically favorable. Not surprising, it's a very strong oxidative process, and oxidative reactions tend to be very energy releasing. This last one notwithstanding. Okay. I'm not going to talk about the glycolysis cycle today. I am going to talk very briefly about this, and then we'll do a brief song and we'll be done. Okay? So, this pathway illustrates some of the considerations of the citric acid cycle. It doesn't include all of them, but it includes some considerations of the citric acid cycle. The citric acid cycle is one that we call anaplerotic. A-N-A-P-L-E-R-O-T-I-C. Anaplerotic. Okay? Anaplerotic means to fill up. To fill up. All right? Now, that's used to describe this pathway because intermediates from this pathway can be used to fill up other things. For example, alpha-ketoglutarate can fill up the cell with glutamate if the cell needs glutamate. Oxaloacetate can be used to make glucose in animals besides human beings. Okay? Oh, I'm sorry, back up on that. Oxaloacetate can be used to make glucose in any organism, but not in net amounts in human beings. Okay? Well, and we'll talk about that. Oxaloacetate can be made into aspartic acid. And conversely, aspartic acid can be made into oxaloacetate. Okay? Fumarate appears in pathways for making nucleotides. Succinate appears in pathways, and no, I'm not asking you to memorize these. All right? Succinate appears in pathways for making heme, something that you need for hemoglobin. Okay? So you see many of the intermediates here are found in other metabolic pathways. Very, very important considerations. And again, many metabolic pathways give breakdown products that feed into here and can then be used. So that anaplerotic nature, that filling up that this pathway does, is a very, very important consideration. Okay. Now, let's see, what else can I tell you? Oh, okay, this upper thing here. All right. So what I was telling you before was you cannot take oxaloacetate in net amounts and make glucose. And another way of saying that is you can't use acetyl-CoA to make pyruvate. If you could do that, you actually could make glucose, but you can't do it. At least mammals can't do it. Bacteria and plants can do that. We can't do that. And we'll see why that's kind of interesting that they can do that. If you could do this pathway up on the top, acetyl-CoA to pyruvate, you probably wouldn't have a weight problem, or you would never have to worry about a weight problem. It would be very easy to burn off excess fat by converting it into glucose, which then you can readily burn. Okay. We can't do that pathway. We have to burn fat separately. Okay. Now, that said, I'm going to finish today with two things. One, I decided to give extra credit today. It's Friday. I figure you should get a reward for coming to class before the holiday. And I thought the jokes went so well last time that I'm thinking this might be a good day for a joke as well. So, and while you're doing that, I shall sing a song relevant to the reason we're doing this today. All right? You can join me in the singing if you'd like. Oh no, I missed my class. Someone ought to kick my ass. Perhaps there is some hope for me. Today her make an online movie. Na 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 na. Online movie. Dr. Kevin's always blowing, telling me I should be knowing all that biochemistry. I hope there is an online movie. Na 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 na. Online movie. Got sweat on my brow. I'm starting to weep. I fire up my laptop. I'm white as a sheet. As Firefox is downloading, I'm feeling neat. 'Cause I just found the online movie. Na 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 na. Online movie. But you didn't get the extra credit.